My name is Hartmut Döhner. I'm a professor of hematology, and I'm located at the University of Ulm in Germany. I'm going to briefly summarize the data that we are presenting here at the ASH meeting in Orlando. And this is the first results on a phase 1b study combining intensive chemotherapy with a new class of agents, so-called Menin inhibitors. And it's the compound from the J&J, from Johnson & Johnson company. It's called Bleximenib. So we combined intensive chemotherapy, so standard of care treatment with this new agent, Bleximenib. The Menin inhibitors are probably the most promising new class of agents currently in development for patients with acute myeloid leukemia. So we are presenting data on 25 patients who received Bleximenib at a dose of 100 milligrams twice daily in combination with the so-called 3+7 intensive chemotherapy. The outcome data are really promising. So we have overall response rate over 90%, a composite complete remission rate of 87%. So this is very promising data from these early data. What is always important is safety, the feasibility of this combination, and there's actually no signal for additional adverse events beyond those that we currently see with the standard of care intensive chemotherapy. There are special adverse events that we know with this class of agents. This is the so-called differentiation syndrome, because this class of agent induces differentiation of leukemic cells. So the differentiation syndrome, it's a clinical syndrome with multiple symptoms like dyspnea, fever, weight gain, and if there are signs of differentiation syndrome, it really needs to be treated immediately. Fortunately, there was not a single case of differentiation syndrome when combining Bleximenib with intensive chemotherapy. The other adverse event is of special interest is a specific cardiac adverse events, so-called QTc prolongation, which was seen in 3 patients, but fortunately only grade 1 or 2, and it did not cause a discontinuation or stopping of Bleximenib. So overall, I think these are very promising data and support or inform the conduct of a phase 3 study that is now about to start. It will be a global phase 3 study comparing intensive chemotherapy plus Bleximenib versus intensive chemotherapy plus placebo. So I mentioned this class of Menin inhibitors. This is a class of agent that is specifically being developed in genetically defined AML entities. That is AML with mutated NPM1 and AML with KMT2A rearrangements. Both entities have been shown to exhibit a certain gene expression signature, deregulated HOX/MEIS gene cluster to be specific, which is caused by recruitment of so-called chromatin complexes which contain the enzyme Menin, and blocking these interactions causes differentiation then of leukemic cells.
If our videos have helped you in any way and you're able to, please consider making a donation to help us continue this important work.
Your gift will go 3 times as far when we reach $500,000 by the end of the year.
Every contribution, big or small, makes a difference, and we're deeply grateful for your support.